In this video, we will be discussing the process of DNA replication. DNA replication is an anabolic process used by cells to duplicate the genetic material of the cell in preparation for cell division. The events of DNA replication are very similar in prokaryotic cells and eukaryotic cells, even though their DNA is packaged differently. We will explore the basic steps of DNA replication, and will then discuss some differences between prokaryotic and eukaryotic DNA replication. It is important to pay attention to the differences that exist. DNA replication is a polymerization reaction, meaning that nucleotide monomers are joined together to form the polymer DNA. DNA replication, like all anabolic reactions, requires precursor metabolites and energy. The precursor metabolites and energy are found in molecules known as triphosphate deoxyribonucleotides. Triphosphate deoxyribonucleotides are nucleotides that contain three phosphate groups linked together by two high-energy bonds. The nucleotide component serves as the monomer to be joined together to form the new DNA molecule, and the high-energy bonds provide the necessary chemical energy needed to join the monomers together. This is different from the other anabolic reactions we examined, where we had separate precursor metabolites and energy provided by ATP. Each of the four triphosphate deoxyribonucleotides differ only in the base that is attached to them. Guanosine triphosphate deoxyribonucleotide is shown at the top of the image. The bottom of the image shows the addition of guanosine triphosphate to an existing DNA strand. To add the guanosine triphosphate to the chain, the last two phosphates are released, thereby allowing the energy in the bonds to be used to drive the addition of guanine to the DNA strand. The same events would occur to add adenosine, thymine, or cytosine to an existing DNA chain. Notice that the new nucleotide is added to the 3' end of the existing DNA strand. This is because the remaining phosphate of the new nucleotide is joined to the 3' hydroxyl group of the sugar in the existing strand. In a previous video, we observed a very basic overview of the events that must occur to replicate DNA. To synthesize new DNA molecules, the strands of an existing DNA molecule must first be separated by breaking the hydrogen bonds between the nitrogenous bases. After doing so, each strand is used as a template to build a new DNA strand. The process of replication is said to be semi-conservative because the new molecules that result each contain an old strand of DNA and a new strand of DNA. Let's look at the process in more detail. We will describe the events as they would occur in a bacterial cell first, and then we'll discuss the differences in prokaryotic and eukaryotic replication. DNA replication begins at a location on the chromosome known as the origin. An enzyme known as DNA helicase binds to the origin and begins unzipping and unwinding the helical structure of the DNA. Unzipping is referring to the breaking of the hydrogen bonds between the nitrogenous bases of the two strands. To keep the nitrogenous bases from rejoining one another, stabilizing proteins are needed. The result of the action of DNA helicase and the stabilizing proteins is the formation of a replication fork. After separating the two strands of the original molecule, the next step is to begin synthesizing two new strands using the original strands as the templates. The enzyme responsible for adding new nucleotides that are complementary to those on the original strand is called Dna Polymerase. There are 5 different types of DNA polymerase in bacterial cells, all of which function in a similar fashion. DNA polymerase enzymes can only add new nucleotides to the 3' end of an existing nucleotide chain. New DNA strands, therefore, are always built in a 5' to 3' direction. The anti-parallel nature of the DNA molecule requires that the two new strands be built in opposite directions in different ways. One strand is referred to as the leading strand, while the other strand is referred to as the lagging strand. Let's examine the synthesis of the leading strand first. The leading strand is synthesized continuously in the 5' to 3' direction as a single-joined strand of nucleotides. To synthesize the leading strand, an enzyme known as primase binds to the parental strand and synthesizes a short-strand RNA molecule complementary to the DNA sequence using triphosphate ribonucleotides. The short strand of ribonucleotides is known as a primer and is important because it acts to provide a 3' hydroxyl group necessary for DNA polymerase III to begin synthesizing a DNA molecule. Triphosphate deoxyribonucleotides are paired with the nitrogenous bases of the parental strand conforming to the rules of complementary base pairing. DNA polymerase III uses the energy to covalently link the adjacent nucleotides together by joining the phosphate of the new nucleotide with the 3' hydroxyl of the existing strand. DNA polymerase III has the ability to recognize errors if a base pair is mismatched and then remove the incorrect nucleotide before continuing with the synthesis of the strand. To finish the synthesis of the leading strand, another enzyme known as DNA polymerase I removes the nucleotides of the primer and replaces them with deoxyribonucleotides. Notice in the image that the synthesis of the leading strand is toward the replication fork. At the same time the leading strand is being synthesized, the lagging strand must also be built. The lagging strand must be built in the opposite direction than that of the leading strand. It is not built continuously, but rather is built in fragments discontinuously away from the replication fork. As the DNA continues to be unzipped and unwound, primase binds to the parental strand, producing a primer for DNA polymerase III, just as we see on the leading strand. What we see differently is that instead of a single primer, multiple primers are built in order to synthesize the lagging strand. DNA polymerase I is responsible for removing all of the primers and replacing them with deoxyribonucleotides. This creates fragments known as Okazaki fragments. In order to complete the new DNA strand, the sugar phosphate backbone must be sealed between each of the Okazaki fragments. The enzyme ligase is responsible for moving along the length of the new DNA strand, sealing the gaps in between the fragments to form a continuous strand of DNA. The additional events that occur in the synthesis of the lagging strand cause its synthesis to lag behind that of the leading strand. Upon completion of both the leading and lagging strands, two new DNA molecules exist, each with one parental strand and one daughter strand. The two molecules are genetically identical to one another. Replication of the bacterial chromosome begins at a location known as the origin. As replication begins, it proceeds in both directions around the circular chromosome. Therefore, it is described as bidirectional replication. Bidirectional replication results in the formation of two replication forks, and therefore two sets of enzymes are needed to complete the replication of a single bacterial chromosome. As the DNA molecule is unwound ahead of the replication fork, supercoils may be introduced into the DNA. Supercoils can cause DNA replication to cease. Cells contain a special enzyme known as topoisomerase that serves to remove the supercoils in the DNA during replication. Topoisomerase cuts the DNA, rotates the DNA to relax the supercoiling, and then rejoins the ends that were cut. Following replication, bacterial cells modify their chromosome through a process of methylation. During methylation, methyl groups are added to some of the nitrogenous bases. Bacterial cells generally methylate adenine and, on occasion, cytosine bases. Methylation of the DNA serves several purposes in bacteria. One function of methylation is that it allows bacteria the ability to control gene expression by turning genes on or off. Methylation of specific sequences can be used as a means to mark the location for the initiation of DNA replication. Methylation of bacterial DNA also aids in the repair of DNA. It is possible during DNA replication for an incorrect nitrogenous base to be inserted into the new DNA strand. Bases that need to be removed due to mismatching should be removed from the new strand and replaced with the correct nucleotide. DNA repair mechanisms depend on methylation to mark the original parental strand so it can be differentiated from the new strand that is not yet methylated. Finally, methylation provides bacterial cells protection against viral infection. Some viruses insert their DNA into the bacterial chromosome when they infect the bacterial cell. Viral DNA is not methylated, and therefore it can be recognized by the bacterial cell, cut out of the chromosome, and then degraded. The events of replication are very similar in eukaryotic cells. The DNA must be separated, and then new nucleotides join to form a leading and lagging strand. There are a few notable differences between the replication of bacterial chromosomes and that of eukaryotic chromosomes that you should examine. First, the linear structure of eukaryotic chromosomes and the number of chromosomes causes the eukaryotic cell to require more than one origin of replication. For example, in a human somatic cell there are 46 chromosomes. Each chromosome would have to have at least one origin. Due to the large size of the chromosomes, they have more than one origin for each chromosome. Eukaryotic cells generally have thousands of origins of replication for each molecule. By having many replication origins, the time needed to complete synthesis is greatly reduced. Another difference is the size of the Okazaki fragments produced when synthesizing the lagging strand. Bacterial cells synthesize Okazaki fragments that are approximately 1,000 to 2,000 nucleotides in length. The Okazaki fragments of eukaryotic cells are much shorter, with only 100 to 400 nucleotides per fragment. Eukaryotic cells also methylate their DNA as a means of regulating gene expression, but instead of methylating adenine, plants and animals methylate cytosine bases.